name is Iho Mehe. I'm an MFA student in creative writing here at UBC and a resident member of Green College. I'm also the academic host for the evening, and I'll start by giving the land acknowledgement. We acknowledge that UBC Vancouver is located on the traditional, ancestral, and unceded territory of the Musqueam people. For thousands of years, the Musqueam community has lived on and cared for this land, and their knowledge and traditions continue to this day. As we gather here, we recognize our responsibility to learn from this history and to engage with the indigenous communities in meaningful ways. We have a truly wonderful event prepared for you today, and I'm truly excited for us to begin. Please welcome Emma, who is the moderator for the evening.
Thank you very much, Homer Hay. My name's Emma Cunliffe. I have the honour of serving as the principal here at Green College, and it's lovely to see so many of you friends, old and current resident members as well, for an event which we designed very much to bring generations together. So it's really great to see you all in the room. In today's iteration of the Emeritus College series, Disciplines Over Time, we'll be focusing on immunology over time. We've assembled a fantastic panel to talk about that topic with you. Let me introduce from your right to left, starting with Michael Gold, who's a professor of microbiology and immunology and about to be emeritus. Am I right, Michael?
Could be.
So we are stretching tonight, but we just about...
I could move there.
Beside Michael, Maria Tokuyama is an assistant professor in microbiology and immunology. And then well known to the resident members, two of our own this evening joining us from the immunology department. Erin Evans is a PhD student, and Tom Worthington is a PhD candidate both in microbiology and immunology. For those of you who haven't attended one of these events tonight, the format goes more or less as follows. We'll spend about half an hour with some set questions that I will pose to each member of the panel, thinking about time and change within the discipline of immunology and how each of our panellists have experienced the discipline, have entered the discipline at a slightly different moment in time, and have understood the discipline accordingly, differently over time. And after we've had the kind of slightly more formal kind of conversation about that, there'll be an invitation to the panellists to chat a bit among themselves before bringing all of you into the conversation. We hope to have plenty of time tonight for conversation, and so as you're listening, if you have ideas about questions you'd like to ask, please save them. We will give you plenty of an opportunity to ask those questions. In each round of questioning, I'm going to begin with Michael and work from there, essentially. And so, Michael, if we can begin with the first question, it's really what drew you to microbiology and immunology when you were considering what you might do?
Okay, thanks very much. It's great to see such a nice audience here. So when I was a high school student, I was not interested in biology at all. I was interested in math and physics. I started college as a physics student, and if all those advanced baseball statistics had been invented then, I would have been a statistics major. My friends were all pre-meds, and they said, you must take this class. It is the best class in the university. It was molecular genetics. And it was just at the right time when in our tutorial sections we were learning about new advances like DNA cloning and how immunological cells worked. So we had this problem. We knew that you could make antibodies, protective proteins against almost anything. You'd never seen it before. How can we make antibodies against anything? If we figure out how much of our genome, how much of our DNA we'd have to devote to that, there wouldn't be room for anything else. And in our tutorials, we learned that the lymphocytes, the immune cells, did it in a very interesting way, very mathematical way. They used multiple gene segments, of which they had many, many copies, and used them in combinatorial ways, so that a small amount of DNA could give you millions and millions of combinations. And even when they put together the gene segments, they intentionally did it in a sloppy way to make more. I said, this is a crazy field. And when else do you get to see a field at the exact time when everyone is going through that everything you knew was wrong phase? And it was really exciting. I decided I had to be an immunologist at that point, and almost nothing was known. And that was, I think, the really great thing about it. And still, when I teach immunology, I used to teach a large second-year class. I point out to the students, this is not known, this is still not known, or some of this was discovered in my lifetime, but it'll be discovered in your lifetime. Maybe you'll discover it. And always at the end of the course, I'd get a lot of teaching evaluations that would say, this course needs a new professor. This one doesn't know any of the answers. But I think the field is transformed in a very interesting way. The way that a lot of fields work. Like you start with making a parts list. You get that IKEA shelving unit. You got 18 of these little dowels. And then you figure out, like, what does each part do with the next part? Okay, you put the dowel into here. I got it. It's a connector. And then you start putting it into modular units. And then over time, you start putting it back into the functional whole. And this is the kind of things now we're doing with animal models and also with patients and manipulating the immune system. So the amount of change and progress in one person's lifetime has just been incredibly impressive.
Thanks so much, Michael. So Maria, we're going to turn to you next. And I wonder if you can talk a bit about what was happening in immunology when you entered the field, and what was it about what was happening that drew you to it?
Yeah, thank you so much. And I echo Mike, that thank you all for being here tonight, and thank you for listening to our different stories throughout different stages of our careers. And so to answer sort of the question of what was happening when I entered this field. So I entered this from not necessarily because I wanted to get into immunology first, but I entered this because as a young scholar, when I was a late high school, early undergrad, I really wanted to study viruses. I read this, virus discovery, virus hunters book when I was in high school, and these outbreak books. This is starting to sound a little bit familiar in this day and age, but really there are these individuals that go out and identify new viruses. But sort of coming from, and what I really wanted to do was really get into this research of how do you study these things. So I really entered this from that perspective and enter into an undergraduate laboratory, my first year in undergrad. And what I found out quickly was that in order to study viruses, you also actually have to understand the host. And what about the host you actually have to understand is how the host actually responds to pathogens like viruses. And that's really when I found out and uncovered and discovered that there is this whole field called immunology, study of the host immune responses and all the players that are involved. And at the time, I think all of sort of the foundation of the immune system, different cell types, what each of these immune cells did was, discovered, and we knew a lot about this, but I think it was still at the surface level. From then going on forward, we really uncovered how expansive the immune system is in terms of its reach and how many interactions does it have with different parts of the body, not even within the immune system, but even other tissues and other places. And I think it's changed quite a bit in terms of our appreciation for how much immunology actually is important for every aspect of our life, and having health and it touches every aspect of health. And so, but yeah, I kind of entered it from a different way. But now I've fallen in love with sort of immunology in a sense that the ability to be able to understand more about how immunological processes work by learning how they respond to external cues. So things like viruses is what my lab studies. And I think there's more to be uncovered there. So, that's what I do.
Thanks so much, Maria. Okay, so Tom has just passed his comps. Congratulations, Tom. It's a really big moment. And so I'm going to start by asking you a definitional question without notice, Tom. Can you please define for us, because I'm conscious that we are quite a mixed audience tonight, what is the kind of remit of the field of immunology, and then talk about what drew you to immunology rather than cognate fields?
Okay, cool. definitely understand that question as well. So what really drew me, I guess at the beginning for me, was a different sort of perspective to the others. For me, it was really like mentorship. During my undergraduate, I thought the immune system was like super cool. I thought it was really interesting. It came up with everything, as Maria was saying. It's implemented in every disease, every system you have in your body relies on the immune system. But for me, it was like this professor that I had in the second year of my course that she taught immunopathology, Dr. Sarah Richardson, and she was just like this amazing professor. She was so cool, she was so enthusiastic, and she loved it so much. And like, she was similar to Mike in the sense that she told us how much we just didn't know, and like how many things were just like, immunologists love making rules and then finding something that immediately breaks that rule, and just like, it was really cool. And then I sort of tailored some of my classes around immunology, and then I did cancer immunology originally, and then I was way too scared by that. Like, I convinced myself I had every cancer that we studied about. And then I found, I was also doing immunology during the pandemic when I was studying it, and so viral immunology I thought sounded really cool. Through like my honours thesis work as well, I came across this idea of like working with viruses and the immune system in the context of autoimmune diseases, and thought they were really lovely like crossover. But yeah, I think that's sort of what drew me to it.
Okay, so lay definition of immunology. You still haven't given it to us.
Sorry, yes. Lay definition of immunology is, well, it's just like the study of the immune system, I guess, in a very general sense. The immune system just being like the way your body defends against foreign invaders and also regulates itself, I guess. Sorry,
Perfect,
yeah.
thank you so much. Sorry to ask you a question without notice.
No.
He passes.
Yeah, he does. Definitely. But both the expert and the layperson. Erin, how about you? What drew you to immunology?
Yeah, so I feel like at school I learned immunology, and I thought it was really interesting, and that was the area where I kind of got the highest marks because I was just so interested in it, and then decided to go to university and sort of like followed this sort of path of molecular biology, so looking into biochemistry and molecular biology, genetics, basically everything that's too small to see with the eye, I was like interested in. And then I did such a weird array of courses at university that they couldn't specialize me. They were like, you've got a degree in molecular biology now. You've done courses in like genetic disease, biochemical disease, microbes. And it was so fascinating to me just because I also read like a deadly diseases book where I was just like, Mum, did you know about cholera? And she's there like, what was this child on about? And it was just really interesting, and I got more into it. And then, yeah, I worked for a bit in sort of antibody research. And so obviously that you know a lot about diseases in that way, but then also antibodies produced by the human body. How can we harness that in therapies? And then ended up in a PhD looking at sort of like viruses and autoimmune disease and the immune system, and it's just like everything I'm interested in. So.
I mean, to me, it sounds like two things. One is, one of the things that often comes up when we ask that question across this series is the importance of a good teacher. And so, Tom, it's really lovely to hear you talk about Sarah Richardson. And Mike, I've got to laugh about the having no answers because I get that from my students too, definitely. But also, I think curious students are intrigued by the fact that there are no answers, and that can really drive a field forward. Mike, I'm going to turn back to you. The second thing that we often hear is that a field can be many things, that people are attracted to a field which is changing, where perhaps the skill set is changing, where there's a sense that there's still good work to be done. And I certainly hear that from all of you. Mike, I'm going to turn slightly. You've talked a little bit about how the field has progressed over time, that some important discoveries have been made and questions have been answered. I'm sure new questions have arisen from those answers. How have you experienced the boundaries of your field to change over the course of your career?
Well, this is really interesting. I think we become much more integrated between different fields. I think when you're first taking things apart, everything seems like something separate. Even in our department, when I first started, I thought of it much as the Department of Microbiology or Immunology. There are people who study the bacteria and the viruses, and there are people who study the things that killed those things. And we hardly ever talk to each other. And then we learned about the gut microbiome. So there's a microbiome, bacterial communities in your gut, on your skin, and it controls everything. It makes small compounds that diffuse into all your tissues. It sets your immune system, it talks to your brain, and all of a sudden everyone is studying the effect of the microbiome on this or that disease. And also when my former students, my students go to medical school, they come back and they tell me every disease has an immunological component. Every disease involves inflammation. And so everything is becoming integrated. Maybe we'll talk about this later. The thing that I teach when I have seminar courses, I'm interested in neuroimmunology, how the brain and the immune system talk to each other. Now, these are two systems that are geared to combating external threats. Why shouldn't they talk to each other? They've been around each other for a very long time. And now we're learning incredible things like your nervous system, all your lymphoid organs, your spleen, they're all innervated. Your nerves are secreting neurotransmitters that regulate your immune cells in some very interesting ways. You know how the fight or flight response, you get a big adrenaline push. Like if I'm going to get eaten by a saber-toothed tiger in the next 30 minutes, I don't really care about that cough, right? So it turns down your immune system in that case. And the immune system talks back. And it's very funny how the way you discover things first, and they get named in a certain way, and it prejudices how you think about something. We've learned a lot about hormones that the immune cells use to talk to each other, and we stopped thinking that maybe they talk to other cells in the same way. So a lot of the immune cell hormones that we've studied, now we're learning that they act on the brain in very interesting ways. So when you get sick, you have this stereotypical sickness behavior. You want to get warm. You want to isolate yourself so you don't infect the rest of your colony or something. And this is all regulated by what we thought were immune cell hormones. So the connections are just enormous. And the kind of directions that I am interested in, since I started as a physicist, now I'm becoming as a mathematical and physical biologist. You know, where do those forces come from on cells? How can we quantify that stuff? So my wife is a cell biologist, and when we talk about, she's also an immunologist, she says, let me draw you a picture of a cell. And I say, let me draw you a graph.
Yeah, so Maria, same question. How have the boundaries of your work and of your field changed over the course of your work?
Yeah. So I think my current research and the research that I've been working on have really sat in really on the boundary. I'm a viral immunologist. I study both virology and I also study immunology. And it's interesting because I think conceptually, a lot of folks like to understand whether I'm in this bin or that bin. Am I a virologist or am I an immunologist? Which one are you? And it's really hard. And it seems challenging, especially with different, maybe it's the naming of departments, and like Mike was saying, naming of fields that you tend to your brain wants to bin you in different categories. But I think, what I found really interesting and exciting about sitting at the boundary is that you actually learn more about each of the fields by having both perspectives. And so I can certainly learn, at least from my research, we can only learn more about the immune system if we also understand how it interacts with viruses that we haven't studied before, right? We can, on the contrary, we can learn a lot about viruses by seeing what kind of pressures the immune system is putting on the virus and how it evolves. And so I think this, we sit on the boundary, and I think there's a lot of benefit to sitting on these boundaries. And as Mike was saying, immunology is sort of turning into a multidisciplinary field. Now we have, connection with, pathogens like viruses or bacteria. Not only that, but we also have sort of engineering that's being, kind of incorporated into immunology. We have a lot of, computer science or sort of AI, extension of bioinformatics that's coming into immunology. Like Mike was saying, neurobiology is also coming into immunology. Evolution is a critical part in sort of understanding immunology. So it is really interesting to actually sit at these boundaries and being able to appreciate immunology from different perspectives. And so we try to value that by having different projects that kind of sit at different sites in our lab. Yeah.
So cool. Thanks, Maria. Erin, I'm going to go to you, Pez, this time. You're, of course, to some extent earlier in your career, and so I'm going to reframe the question slightly. How are you imagining the pieces that you have to bring together to do the work that you want to do, both for your PhD but also moving through a potential career in immunology?
Yeah, I feel like, because when you originally posed this question to us, I was thinking about how my undergrad was quite interdisciplinary, and I was trying to think about a lot of different topics, all say within the cell, but fully understanding the cell. And then what I study currently is I'm looking at how viruses are affecting the development of MS, so multiple sclerosis. So that's the involvement of the immune system and neurology and viruses. And that's something I really like, is being able to like dip my toe in lots of different fields. But it can be quite overwhelming sometimes, because you just look at everything that you need to know. I'm sure you have this for your comps, and you're just like, how am I ever going to know that much? And I sometimes get jealous of sort of Mike's generation, where I'm like, they just had to know that like there were these two cells, and that's it. And
Passed my comps, yeah.
yeah, exactly. But yeah, so I guess what do? I look at animal models, and I'm doing cell, like immunotyping of the cells, anatomy as well, like the different regions of the brain, different like structures of viruses and proteins and genes, and yeah, lots of stuff.
I feel like I've tapped into your deepest fears. I'm sorry. And it's just to reassure you, I think it's true in every field that we look at other people and think they know all the things and I know none of the things, but the truth is in any field people have overlapping knowledge, and that's what makes the field strong. Tom, what about you?
So in the sense of what I have to bring forward, is that the idea?
Yeah, in the sense of kind of thinking about what you'd like to achieve with your work. What are you drawing from, and what are the things that you see as being important to allowing you to do the work you'd like to do?
Yeah, I guess when choosing my PhD programs and choosing what I was going to study, I definitely wanted to do immunology, and I thought cellular immunology was just like the level of science I was really happy with, the techniques and everything like that. I also had this really big fear of genetics for some reason. I just hated it, and so I avoided it like loads. And I also didn't do any clinical trial stuff or do anything like clinically as well. And so what I definitely discovered is like doing my PhD and like studying immunology. I mainly look at like new therapies and developing them and then applying them in the clinic. And for me to actually help develop these therapies in mice, I actually have to understand like how they're applied in clinic. Like I've had to learn how clinical trials work, how the therapies are applied, how they work in humans, how there are differences between animals and humans, and so it's required me to have a lot more of a holistic approach in these ways. Also, calling immunology just immunology, as we were saying before, isn't true. Like it's like everything, and so like I have to understand the genetics behind the immune cells. I have to understand how it interacts with the brain and how it interacts. So I've had to go into so many different areas since starting my program that it's been really fun. And I think in terms of like learning that, there are so many online resources through UBC, through online courses. I think I'm registered at the University of Penn State for some things. Like it's just, you have to just like dip your toes into so many other things, because if you don't, then you're producing research that's like very narrow-minded and actually not applicable to what you're trying to do.
No, it's so good to hear you talk and think about understanding functionally, for example, how things work at the cellular level might not work at a genetic level, and that may be a kind of a foundational stumbling block. And so having to have that cross-sectional understanding, that cross-disciplinary understanding. My last question, fun question. What's the most exciting thing that's happened in the field in your time, in and around it?
Oh, there's so many. I think different perspectives. So when we learned that the immune system used all those combinatorial approaches to make completely random receptors against things we've never seen before, so we're protected against everything. My PhD was saying we've evolved in the presence of bacteria and viruses for millions of years. We know what these things look like. There ought to be like receptors that are evolutionarily conserved for those. And they are. And I didn't discover it during my PhD because the technologies weren't there yet. But they were found first in Drosophila because they were fruit flies, because they were evolutionarily ancient, the way that ancient organisms protect themselves against bacteria and fungi. And what I've liked is how enabling technologies have really changed the field. You know, as someone who had a physics background, my always view is like you come up with an idea of something you want to study, and then you decide what you want to measure, and then you build the machine to measure it. And the things that have changed that have allowed us to do things that had clinical implications, I think this has been the best, the biggest discovery. So monoclonal antibodies, being able to make an antibody that recognizes only one thing, being able to make it in culture or in bacteria, so you can make an infinite amount of it. We can now use these things clinically to kill cancer cells, to allow your T cells to go on for longer, to kill the cancer cells, to block harmful hormones, so inflammatory hormones that cause rheumatoid arthritis, we can block these with the monoclonal antibodies. So the time scale, I think, of going from discovery to clinical translation is getting much, much shorter. And this is really exciting. Why?
Thank you, Mike. You're so generous with that, Mike. It's good. Maria, what about you? What's the most exciting thing that's happened in your time in the field?
Yeah, I think it's like Mike said, it's hard to pick one, so I might cheat and say two. And so one is technological advancement. In immunology, we care a lot about different types of immune cells that do different things that is important for health. And it used to be that you would look under the microscope and look at the shape of the immune cells, you know, and they say, oh, these are round and small, whereas these are bigger and they're amorphous. And that's literally how people used to define different types of immune cells. And since then, I kind of entered already at a stage of my career when you were able to definitively say what proteins these immune cells are expressing. And by understanding what proteins these cells are expressing, we can actually monitor them through a really fancy machine called flow cytometer that allows these antibodies that Mike was talking about that target the specific proteins on the immune cells, and you can tag them with different fluorophores, so different fluorescent markers. And when I started, you could only look at two different colors. So if I had two different proteins, I could only look at two different proteins on the cell. And now, since then, now we're able to look at, over 50 different proteins simultaneously, all on the individual cell together. That's just using one technology, using the flow cytometer. But we also have this mass spec-based method that allows you to label over 100 different proteins on a cell surface, or even intracellularly inside of the cell, and look at all of them in a single experiment. And even beyond that, we have this sequencing technology that allows us to then monitor thousands of genes that are just expressed in individual immune cells that allows us to see this at an even higher resolution all in one go. And so I think just the resolution of the amount of knowledge that we can gain now has been tremendous. And so I think that's really revolutionizing how we think about immune cells, identification of new immune cells and what they do. And so that is one. So sorry. Second is that really now that we understand how immune cells work at the basic level and at many levels, we can actually modify them. So we can actually make use of that and take that, use that to our advantage and engineer immune cells to do the things we want it to do. So if you have cells that are actually not, immune cells that are not working well, we can actually rearm it to do something that we want. We can also target cells, immune cells, that normally won't target the cancer cell that we really wanted to, but now we can engineer these cells to be able to target those. So these are some things, both of these things we're doing in the lab, and I think it's a really exciting time in immunology to really apply all of the knowledge that we have for new therapeutics and new developments in the field.
Can I please ask you a follow-up about your first exciting development?
Yes.
Can you explain, for those of us who are not in immunology, allowing yourself to see a greater number of proteins in a given experiment or to express a greater number through spectrometry, what does that allow you to do that you couldn't previously do?
Yeah, so before the number of, thank you for the question. I think this is really important. So number of things. So one is we can actually see that in fact, one type of small, singular, really round cell we called T cell in the past. Now we know that there's many, many different types of T cells, right? So it's not actually one homogeneous group of T cells, but we actually have many different types of T cells that all have various functions. And that's only one immune cell. Now we can go to B cells, the same thing there. Dendritic cells, same thing. So we have actually identified over the course of maybe 20 years a huge number of new immune cell types that we didn't know before, because we can actually characterize them by what kind of proteins they express and how they function. And secondly, we can really understand, not only what they are and the different kinds of immune cells we have, but what they do, right? So the gene expression, the sequencing technologies allows us to say, in this context, this particular subset of T cell expresses these sets of genes, and now we know that this is really important for fighting pathogen, while this other tiny subset of cell that expresses these genes are actually very detrimental in chronic disease. And so all of these characterization and phenotyping that we can do is really deepening our understanding of how our immune system works.
You want to jump in?
Oh, no, that was an analogy I used in the second year class. So in the beginning, you said we only had two colors. So if we were doing a flow cytometry analysis of this audience, in the beginning, we could only divide you into people who were wearing blue shirts and people who weren't wearing blue shirts. And we'd assume that everyone wearing a blue shirt was identical. So now we can look at who's wearing a blue shirt, who's wearing red socks, and onward from there to many different parameters, and discover that you're actually all different.
Yeah, that's a great metaphor. Thank you.
People wonder why we're overwhelmed as PhD students. There's just so much to know.
Erin, I'm going to go to you. What's the most exciting thing that's happened in your time in and around the field?
So I think for me it was CRISPR, which I feel like has been, and everyone's nodding at me, yeah. And so it I first heard about it when I was sort of like at the end of high school, just getting into university. And CRISPR is sort of what we call molecular scissors, so it allows us to go in and cut DNA and we can chop it out and then we can put something back in. And it's been really useful for animal models. So one of the downsides of using an animal model is that it's obviously not a human, but we can put the human gene into mouse models, and that allows us to we're more able to look at the human gene in the context of disease or infection or whatever's happening. And then not only can we do this in animal models, we can do this in like cell lines, and there's many, many different applications of it. And then you might have also heard about like CRISPR on babies, which obviously gets into a whole sort of like ethical debate, but there is hope that CRISPR will hopefully be like a therapy in the future. And I think CRISPR is now just utilized so often to help answer questions that we have. What does this gene do? Knock it out, see what happens to the mouse, and you have your answer maybe in a few years.
Very cool. Thank you, Tom.
I guess maybe very similar to what Maria and Erin said. I think our ability to actually directly manipulate immune cells to target cells we want to target, even host cells, is crazy. And we're already seeing the implications in therapies and cancers like lymphoma that was once death sentences and now have some of the highest survival rates due to the fact that we're able to trick our own immune system into targeting these cells, basically. I guess not maybe a technique, but more a theory that I thought was so cool is this whole idea of the hygiene hypothesis, which I love loads. And it's just, I sort of came to it during my undergrad degree, and it's this idea that the increased sterilization that we grow up with our food and our drinks and our lifestyle has drastically changed how we interact with environmental microorganisms. And as a result, our immune system is just so confused on what to do, and it's kind of panicking a little bit, and we're seeing this massive rise in these inflammatory and these autoimmune diseases and these really debilitating diseases that are affecting so many people. And another thing that's really sparked my interest in it is because we're only really seeing the implication of these hygiene practices in our generation, me and Erin's generation. We're seeing huge rates of IBD and type 1 diabetes, and it's associated with these sterilization techniques. C-section's a very famous one that's very current at the moment. This idea that actually if you're born via C-section, you don't get your mum's microbiome, and then your immune system doesn't interact with these important microbes when you're born. And children who are born through C-section have major increased risk of certain diseases, 25% higher risk of type 1 diabetes, of celiac disease, of asthma. And it's just this idea of this slight change in the first hour of your life has such debilitating impacts. And yeah, I think it's just crazy, and the immune system's stupid, and it's so much fun to learn about.
Thank you. Those are such great answers because they're so different as well. So thank you. Okay, what we're going to do now is to spend a few minutes giving the panel an opportunity to speak to one another a little bit. Tom and Erin, I'm going to start with you. Are there any questions that you've always wanted to ask Maria and Mike? And now's your chance.
I have a question, actually. If you weren't allowed to study immunology, what other area would you have studied? And you can't say virology.
I already talked about it, baseball statistics. They invented all these new statistics that I didn't know what they were about. I still don't understand them all. How many times you're on base, your value above replacement, how much better are you than the average player at your position, how they quantify that. When I retire, I'll go study that. Yeah. There's lots of things I want to know. I want to learn everything. So I used to, when I would teach the big 300-student class, it was an hour-and-a-half class, I'd take a break in the middle, and I said, when I retire, I would like to learn more things. I'd like to learn languages, all of them. That would take a while. And I'd ask the students, what are the best courses on campus that I could sit in on with permission? And I would like to learn all kinds of stuff: history, archaeology. There's so much: music, more art, things I haven't studied as science. Yeah.
All right. If it wasn't immunology or virology, right? I mean, I guess maybe going from the next closest thing and then the further out thing. Maybe evolution, getting really more into evolution. And I think that actually touches on our work as well, because in my lab we study these ancient viral sequences that are embedded in all human genome. We call these endogenous retroviruses, and these are sequences that we say, this is what I say in my introductory slide at every presentation, these came from our primate ancestors, and these have been in the genome for 40 million plus years. And I always have to stop, even though I say that in my introduction, I say, what does that exactly mean? How do we get from there to here, and how do these sequences get to where we are? It is a really big jump, and so there is a field who studies this to study this particular question, but I would love to get more into it and further into my career and say, how do we get this? How do we track some of this evolutionary history of these viral sequences in the past, right? And then that kind of gets into sort of like maybe archaeology or maybe some, something like this. And then I think it's also interesting to understand more social science, like behaviors of people, and even connection of how does our, health and what we study at more of a cellular molecular level actually affect behaviors, social behaviors, and can we learn more about how people do what they do and why they do them from using kind of like our more molecular and cellular knowledge. That's pipe dream, right? But, yeah.
Go ahead. Yeah.
If you had an unlimited budget and like unlimited resources, what's the question you would want to answer?
You're first.
And first, so we are really saying unlimited budget, right? Okay, we're
If my
really.
PhD had unlimited budget, what would you make me do?
And is there a question of feasibility or access to resources? Okay. I think this is something that, again, it comes back to what our lab is really interested in understanding, which is what makes individuals different and how they respond to pathogen. And so why does, this, and we saw this during the pandemic actually, right? Some individuals will have asymptomatic disease and other people will have very severe disease. And there are many factors that went into the differences in disease outcomes that we also already know of, right? Age, comorbidities, sex, all of these things. But there are other, sort of far beyond what we can actually see, individual differences that contribute to these different disease outcomes. So if I had unlimited budget and unlimited, feasibility is not a question, I would love to be able to survey sort of the immune status of as many individuals as I could possibly get my hand on, and understand, what are there patterns that we can infer from characterizing the immune responses of a huge range of people, and figure out what makes them different about how they develop disease, right? And that could be depending on host factors that that we study, or it could be environmental factors. But I think just having this massive amount of data that we could dig into would be what I would do. Yeah. Right.
I think that goes more into like how biology has become like big data lately. We have many more sort of like samples, and bioinformatics allows us to look at way many more n's, many more numbers than we've ever looked at before, and we can get these better answers that are more representative of the whole population.
That's exactly right. But I'll go to the other end of the scale. So I'm sort of a mechanism guy. So I would like to be able to look at a cell and have every cellular component labeled in a different color, and have a microscope that can watch them all in real time, and probes to look at things like forces between things, look at interactions to see what interacts with what in real time, over a long time scale. And then take it up to the animal models. Now we have really cool things, like we can do what's called 2-photon intravital microscopy. So we can modify cells in a mouse so that like maybe the one type of immune cell is green all the time, one type is red all the time. And it's hard to do microscopy in an animal because the wavelengths of light that you use to excite the fluorescent molecules don't really penetrate. But physicists came up with this way of doing 2 photons. You use 2 different wavelengths of light. They both hit the same electron, excite them. Now you can look at things inside an animal. So people often look at something thin, like the ear. And you could look at, start an immune response, and look at the T cells and B cells and all the other immune cells migrating in and doing their thing. And I think being able to look at things in real time, in a whole thing, not having it taken apart. Like we like to see how the whole car drives, not like just how like this part works. I think that would be incredible, and we would need big data. We would need the world's biggest computer to understand what all this does. So we do some of these things already, like we do super resolution microscopy. We can look at individual molecules, and we take snapshots, and we would take like 40,000 snapshots over like an hour, each with hundreds of spots. So every sample is like millions of data points. And like, obviously we're way beyond pen and paper, what I can do with those baseball stats.
Questions of the evening right there. Okay, Mike and Maria, your turn. What questions would you like to ask Tom and Erin?
So in your career, you're going to move to different levels. You're going to finish your PhD. You're going to do a spectacular PhD, both of you. Make many new discoveries, and then maybe you'll want to go on to another lab to be a postdoctoral fellow, where you could expand on that, or you could do something completely different so that in the future you could combine like two different unique skills to make your own scientific niche. So maybe that's a little far in the future. Let's start about the next step. Let's start planning your postdoc. Like, what do you think you might want to work on? It could be anything that's kind of intriguing, related or not to what you're doing now, and maybe you want to go to some exotic place to study it. I don't know. Baltimore. I don't know.
I try not to think of my postdoc. I guess from one of the courses I took with you, Mike, the sort of impact on like childhood immunity is crazy. Like we know nothing about the first, very little about the immune system of a fetus or the immune system of a newborn. Obviously, they're very hard populations to study. But I think it's just crazy how their immune systems are so weird and just non-functional and just completely opposite to our immune systems now, but they're also so good at the same time and they just work differently. And I think maybe there's just loads of questions there. I think that'd be really good. I think the next exotic place for me to go to is probably London back home. So I think, going back there for a bit as well, just to see what it's like.
Thanks. I don't know. I think so my undergrad I did oral pathogens. My master's I did Drosophila. My a job I worked in antibody development. So I've kind of done a lot of random things all over the shop. But what I really like is when it all kind of meshes together. And thank you. And I think my ultimate goal is I'd like to have a lab, and I'd like us to study kind of like autoimmune diseases I'm very interested in, but if we cure them or we fully understand them, I've got to have something else to look at. But I think just this interaction between humans and pathogens and sort of and it all comes back to the immune system. Luckily I've got a long time to figure out my niche.
All right, so I guess now that you have both been in grad school for a couple years now, I guess was there something and you've been doing, experiments, developing your projects, and so is there anything that you have noticed in the last 2 years that you weren't expecting at all, whether it's, the process, your journey, or scientifically, or being a full-time grad student for the last 2 years studying immunology in both of your mouse models, right? And so I don't know if there was any shock element or something that you weren't expecting, Not really? Well, I'm thinking
All your experiments work all the time.
Well, I guess I already knew that not all my experiments would work all the time. I think, I guess I knew about it tangentially, but knowing that I have to communicate my research to both the layperson and scientists, I have to talk to Mike about my research, and I'm like, oh my God, he knows way more than me. If I'm talking to my committee, I'm thinking, why am I here? They know way more than me about my project. They can just get their heads together and they'll think about it. And so I have to do that quite high level making sure I cross all my T's and dot all my I's sort of thinking. And then talking to lay people because I study multiple sclerosis, and so there's a really big engagement of the community of people living with MS, which is amazing for our development as scientists and sort of like the field in general. And part of that is being able to communicate what I'm studying to people living with MS and my family and people who know nothing about immunology, because what we study is so important for, like, everyone has an immune system, and if you don't have an immune system, you need to know it even more. And so, yeah, just that sort of like communication, and people think scientists are just in a lab, like, pipetting things, but we actually have to be quite engaged sort of socially in communicating, and yeah.
Yeah, no, that's a really good point. I think the other one for me is just the amount of like teamwork is involved in research and how it's both independent and so dependent at the same time. Like, my lab manager made a comment the other day about how she sees me more than her children, and because like and it's like so true in the sense that I spend like 8 to 9 hours with her every day. We sit next door to each other on a desk, and I see her like 11 months of the year, and it's just like spending for like 5 years as well. And so I spend so much time with these people that I had no involvement in arranging who these people would be over me. Hi, Cynthia, as well. Yeah, and I just how much like it's like speaking with siblings and stuff in your lab, and like how you have to like work so much on how you communicate with these people, and how much teamwork is reliant, and how much you have to work together to make it a smooth operating system. And like, I guess, yeah, I just like in my undergrad experience, research was so independent, and you just had this little project and you had to go into the lab, but that's like not what it is. And it was a very happy surprise. Like, I've enjoyed it a lot, like the teamwork aspect of it, and you never really feel alone. And I think that's definitely a bigger part maybe in immunology compared to other fields. Like, speaking to other people, it's a lot more like other areas are a lot more independent. But yeah, I say that.
Thank you so much. Homer Hay, I'm going to hand over to you to close this out.
Thank you so much for joining us today, and we hope to see you at more of our functions in the future.